Good morning, students. Students, today I want to discuss about the first practical of biology, and this practical is basically to prepare a temporary mount of onion peel. Now, today, with the help of some slides, I will tell you that how a temporary mount of onion peel can be made in the laboratory and how can we observe a cell through it. So initially, if we have to take if we have to prepare a temporary mount on a slide, so we have to take some onions, and we have to cut these onions, and we have to cut these onions, and after cutting onions, we have to keep them into watch glass. Here you are observing four structures which are made up of glass, and these glass are called as watch glass. There are four watch glasses are there. In the first watch glass you are observing a onion piece is kept, while in the another watch glass water, fresh water is there, and here in the third one watch glass you are observing a red color solution is there. This is called staining solution or a dye. This is basically safranin. You must have to remember one thing, that while we are going to prepare a slide of plant cell, then we have to use safranin. This red color solution which you are observing here, this is diluted safranin, and while in the fourth one watch glass there is once again water. So now how to perform experiment I am going to tell you. Initially uh, we have to take a onion, and then we are going to cut it. In the second slide you are observing here. Now after cutting it with the help of a forceps is used to pull out the peel from the onion. Now you are observing here, I am zooming it, you are observing how the epidermal peel is pulled out from the onion. Okay, now here you are observing, now here in the next slide you can easily observe that now this peel is completely removed from the onion bulb, and now you are observing how we remove it from the onion. This is the inner surface of the onion peel, and this is basically made up of epidermis cells or epidermal cells. Now we are pulling it out, then we are keeping it into a watch glass, and you have to uh, take one thing in your mind, that you have to immerse this peel into water, because it will prevent the peel from drying up. Okay, or it will prevent it for becoming dehydrated. Then after it we will cut this peel into the small pieces. Now because it's a large peel, now we have to cut it into the small pieces. Then we will remove it from the water, and we will place it into the saffron in solution. Now here you are observing in the second one watch glass, red color solution is there, stain is there, dye is there, and it is called safranin, and now we are placing it into the safranin solution. Now we will leave it for 2 to 3 minutes, or we can leave it till 5 minutes. Then once again what we have to do that, after removing from this stain, okay, now we will place it into the another watch glass, and which is also containing water. So extra stain will be removed from there. You are observing here in the this one slide, you can observe that the we are just placing it into the watch glass which is having water, and the extra stain is going to be removed from this one step. Now what we have to do, now we will with the help of a brush we will take out the onion peel from the water solution, and now you are observing we have a glass slide, and we have to keep it into the glass slide, and we will keep it we will keep our material, or we will keep that particular peel into the slide, and we will spread the whole peel. We have to keep one thing in our mind, or we have to remember one thing, that the peel should not be fold. Okay, we have to spread the peel on the slide clearly. Now you are observing here, now we have to take glycerin. Now we will use, or we will put 2 drops of glycerin on our material, or on our peel. Are you observing it? Now what is the next step? Now we have a square shape small glass structure which is called cover slip. Now we have to place this cover slip on the slide over the uh, onion peel, and for this process we have to use a needle. Okay, with the help of the needle we can place a cover slip over onion peel. Here you can see that now we have placed it over the onion peel, Now what we have to do in the next step? Now you are observing some of the glycerin is coming out from the uh, corners, or then we have to remove this glycerin from the slide. Now we will take a tissue paper, and with the help of this tissue paper we slowly we will remove all the glycerin which is coming out from the cover slip. Okay, and one more thing we have to take in our mind, that while we are placing cover slip over the slide, we should have to remember that there should be there should not be a single air bubble on it. Now we have placed it and we have removed the extra glycerin which is coming out. Now in the next step you are observing, now we have to take out a microscope, and we have to place this slide over the microscope. Now we will place this slide on the microscope. Now if we will set the microscope, and the eyepiece lens you should have to use 10x, and the nose lens, the nose piece lens, it should also be 10x. So 10x into 10x, it will become 100x. So you can observe 100 times magnified image on the microscope. While you will observe in the microscope, so you will observe a honeybee comb like structures. There are some rectangular structures, these are the cells, and inside the cell you are observing the dark line, or while you are observing a cell, these dark lines are basically cell wall, and in between there is a point like structure, and this structure is nucleus. So while we are going to prepare a slide, we are going to prepare a temporary mount. So I am just very quickly I am repeating the steps. We have to take out one onion, we have to cut it into pieces. Now from one piece, from its inner side we have to remove, with the help of forceps we will remove the epidermal peel, and we will place this peel into water. Then after placing into the water, we will place into the safranin, then we will wash it again, then we will place it onto the slide, then we will put 2 drops of glycerine, then we will place a cover slip over it, and then we will observe it under the microscope, and finally we are able to see such kind of structures. So I hope so you understood very well that how can we prepare a slide in the laboratory. Now you are supposed to write all these content in your copy, okay, and draw the neat and clean diagrams. Still you have any query, so you can ask me. Thank you. Bye bye.